Okay, this is good.
Okay.
So it's my pleasure to introduce Cameron, a young, talented guy from Louisiana, University of Louisiana at Lafayette. And he will talk today about modeling lockdown, contact tracing, and, and testing in COVID-19 outbreak applied to China and United States. So please, Cameron.
Thank you, Pierre, and thank you to the other organizers also for inviting me to this webinar, and it's a really useful platform. So yes, I'm really glad to actually go after Quentin gave his talk, because I'm dealing with a lot of the same issues that they tried, but I'll kind of give a somewhat different approach. So first, let me just make some acknowledgements. So first, we're funded by NSF for this work, and this is also a team effort. So the work that I'm particularly going to be talking about today, two works, are in collaboration with Dr. Hayriye Gulbudak and then her graduate student, Caleb McDonald, both from University of Louisiana, Lafayette. But just to acknowledge the other part of this NSF project is dealing with phylogenetic models, and that's with the UF Emerging Pathogens Institute, but that's still in preparation and ongoing work. So let me just get started. And so I think that the challenges in modeling this emerging outbreaks was somewhat addressed and quite well by Quentin, spot on with that autonomous SIR models assume exponential growth before susceptible depletion, but that's not what we observed in COVID-19 or many emerging outbreaks. And so the problem is that the parameters are not fixed. Behavior of the population changes, control measures are implemented. So these are all challenges for how then can we just transform the traditional SIR approach to fit the data. So, and there's issues with prediction and fitting data, as we know. But I think some important questions, at least that we wanted to address, were not necessarily about predictions here, was how to use the data and some model structure assumptions to get at the efficacy of interventions. And so that's what I will focus on. So I just want to bring up sort of the, challenges that, we first sort of, with Dr. Glenn Webb from Vanderbilt University, we started to work on modeling an outbreak, and that was the Ebola outbreak. And then there was, of course, a large effort among the mathematical biology community and for this West African Ebola outbreak in 2014, but we started to notice, all of the problems there with the modeling forecast. So, this is kind of derived from that work. And so, here's just the question that we sought to answer in that first work, and which motivated us for the COVID, is how to model and quantify the impact of contact tracing, which was the most important control strategy for Ebola, and also a control strategy used for COVID, but to maybe a little bit less effect, and or at least it's much harder given the characteristics of the disease. But the kind of questions we can answer is, given the type of data that we had for, let's say, the Ebola outbreak, where the WHO was reporting for several weeks the percentage of the reported cases that were known traced contacts, we were able to sort of derive from that efficacies of the contact tracing for the different countries and show the actual impact. as you can see in the simulations C and D, Sierra Leone had a much more effective contact tracing program, as the red is the actual sort of cumulative cases, but if they didn't employ their contact tracing, which we estimated from the data, they would have had a very large outbreak. So they had a larger effect, so they're a better effective contact tracing program. So this is kind of the, what we were interested in, and to incorporate contact tracing, and more generally, when we talk about interventions or population behavior change, how do we incorporate these into models? And so we think, so here, I kind of want to separate, I like the title of Quentin in terms of phenomenological model. So we will get to a phenomenological intervention for COVID in a second, but I think of contact tracing as a mechanistic intervention, in the sense that, okay, we sort of, the process of contact tracing is that when there's a reported case, then there's an investigation to track down that person's contacts and then try to prevent them from carrying onward transmissions. And so we can sort of model that as a mechanistic action. So if we consider the cumulative reported cases coming in, so that's equal to the rate of reporting of infectious individuals. But the contact tracing process, again, is going back through their infectious period. So we're integrating backward in time here to identify their contacts. And so we have some probability or some proportion phi here of their contacts, which, here we're interested in the contacts that actually resulted in infection. And so we can track that. And so, okay, it's a very with this setup, it's complicated because it's an integral differential equation. But if we make some simplifying assumption that the susceptible remains constant over the infectious period, then we can just see that the actual number of the contact tracing is related to the transmission. So it's just, you can incorporate, a simple way of incorporating contact tracing is that the fraction of traced contacts just is a multiplier of the transmission rate. And so that, and this is then how we sort of think of the mechanistic action of contact tracing. So again, this is a reactionary intervention. And then for COVID, these were the kind of interventions that were large scale during the pandemic, especially at the beginning. So, but here for COVID, of course, in addition to contact tracing, there was large scale lockdowns and self-quarantine measures. Okay, we're kind of using these words synonymously. Here, we're not saying quarantine of infected individuals. We're thinking of just more so just individuals just sort of at home quarantining, or stay-at-home orders, things like that, or business closures. So that's going to be a more we want to model that. Okay, that's a little bit more of a phenomenological intervention, but we think of it as reactionary. As governments and individuals react to climbing cases, there's sort of this response that happens, seems to happen. It doesn't happen just when there's no cases. So it's somehow dependent on the number of cases coming in, and it strengthens this. Then there's sort of an urge for this lockdown or self-quarantine to more and more happen. So we develop a SIR time model, which I'll show in a minute to try to account for this. And then we're going to apply that to first, because we have contact tracing data from China also, which was nice. We sort of do a comparative analysis between contact tracing and lockdowns in China. For the US, there wasn't really any contact tracing data or much of a contact tracing effort, but we have different questions, which I will get to in the second part of the talk. So the full model that we come up with, it's a little bit complicated. There's more compartments. We essentially want to divide the population into sort of regular normal business interactions. So that's just S, E, and I, where people are not either practicing self-quarantine, or which is the Q state, or being contact traced, which is the C state. Okay, so we have that people susceptibles transition into this self-quarantine state at this rate, or actually proportionality constant, sigma, and we assume it's proportional to the force of infection, because we think again of the self-quarantine or lockdown behavior as reactionary to the cases coming in. And so the simplest phenomenological way we're going to incorporate it is just by being a proportion, the rate as being proportional to the force of infection. Contact tracing, I already showed, mechanistically can be shown to be proportional to the force of infection. And we again use this phi as the proportion of contacts traced. And then there's some other parameters that come up that we use, that in this full model, such as the average length of the individual's self-quarantine state, or alpha C would be the how long that they're monitored for contact tracing. And then if it's imperfect, there might be some susceptibility and infectivity in these different states. But this is a complicated model. So we want to look at a simpler case and to derive some analytical results. So what we can assume is that these states perfectly prevent infection, susceptibility and infectivity of the individuals. And so we call this the perfect quarantine case, both contact traced or just self-quarantined. And then we can write this reactionary intervention, the total susceptible quarantine factor, which we call psi. So this includes both the susceptible individuals that are contact traced, and that's equal to 1 minus P over P. So P here is the probability of transmission upon contact. And so here we are breaking down the transmission rate in terms of probability of transmission upon contact and contact rate, because contact tracing is inevitably involved with the contact rate. And then we have the sigma is this phenomenological self-quarantine rate. So this is the total susceptible quarantine proportionality factor that's proportional to the force of infection. And we make some other assumptions that a sort of indefinite lockdown, and again, the perfect quarantine. And then we get a very simple reproduction number, just the transmission rate times the infectious period, capital T. And then again, the phi here is the proportion of contacts traced, and we're assuming perfect sort of contact tracing. So that's why it's multiplied by 1 minus phi. And in this model, we can derive the final size of the outbreak, and also peak size. And so what we observe, okay, if we think about the traditional formula for final size of an SIR model or SEIR model, what changes when we have this, this total susceptible quarantine occurring proportional to the force of infection, is we get this inverse proportionality with that rate size, or or factor sigma, or sorry, psi. And so this is what we observe. And so we say that outbreak size and peak size are inversely proportional to this population quarantine factor, or more sort of informally lockdown speed. And we can, with these analytical formulas, we can calculate sensitivities with respect to these main parameters. And we see differences in the contact tracing intervention and the self-quarantine intervention. Again, self-quarantine has this simple inverse proportionality, whereas contact tracing, there's differences between the peak and final size, in that for the peak size, we get that the sensitivity, actually, the peak size is sensitive to contact tracing. You can look at this formula and see that, or you can look at the picture, the graph here, and see that we get a good slope. So it is sensitive to the contact tracing. Whereas the final size is flat. It's very flat when there's contact tracing is inefficient. Okay, or there's not a lot of fraction being contact traced. So in essence, basically, contact tracing really doesn't affect the final size unless it's very very perfect or very efficient. Okay, and so this is going to play a role in what was effective in COVID. So we do get a more complicated final size formula in the full model, in a special case of it, when we assume, again, that relax the assumption of the perfect quarantine state, and assume some uniform susceptibility, along with there can be infectivity of the quarantined and contact traced individuals. There is one assumption here that makes it not as tenable as the full model. We do sort of have to make some transition assumptions on the transitions in order to derive this formula, but it does allow for sort of a lower, it gives actually like an upper bound sort of the actual final size if you assume more reasonable transition rates. I'm not going to get too much into it, but we can derive. So this was a nice formula just to derive analytically in a more general case. And it does reduce to the simple formula we observed when we assume, again, that the susceptibility is 0. And then if we don't worry about the initial condition, then we can get rid of the initial condition part of this formula, assuming at the outset of the outbreak. And so we can do sensitivity analysis, again, just to see the effect on the outbreak size. And so here, and this is in the full model, and when we look at different susceptibilities, we see that the actual simulated becomes more off from our formula as the susceptibility of the quarantined individuals increases. And again, that's just because we're assuming here the actual more realistic assumptions in our general final size formula was for a more special case. But we see, again, this inverse proportionality, even on, we can observe it with the log scale of the final size with respect to this self-quarantine parameter. And it can be translated into the time sort of lockdown. So we can think about the percentage of the population that is self-quarantined. And so in the time it takes for 50% of the population to be self-quarantined. And these parameters, actually, I'll show you, were estimated from China. So I'm going to show the data fitting here in a minute, but it just shows you that it's very sensitive to the timing of the lockdowns. So basically, if we look at the time to 50% of the population being self-quarantine, then the amount of cases can dramatically increase as we delay it, beyond 2 weeks or 14 days here. We go over to our other curve, and that corresponds to a very much higher final outbreak size than if we have the time for 50% of the population to be locked down or self-quarantined. If we have it more in line with a week, then, or a week basically, then it's after January 21st, 2020, then we get a much smaller outbreak. And so the lockdown speed was instrumental in reducing, and the final size. The contact tracing didn't have nearly as much impact, we see, because you would have to trace so much fraction of contacts to make any headway on the outbreak size. But it does contact tracing did have an impact. So our estimated, for all of China, contact tracing rate did reduce the peak size. So how did we use the data? Okay, so this was where we used the contact tracing data in addition to the reported case data. So we basically had data from the National Health Service in China, or online, about the daily total quarantine contacts in China. So that's the blue here, dots here. And so that was a computable parameter or variable, in our model, just a combination of some parameters and variables. And so we fit that simultaneously with the daily case counts. And we noticed that we can really break up China into two parts: Hubei, where Wuhan is, where the outbreak originated, had the bulk of the cases, and then the rest of China, they had different characteristics. So we fit, we sort of, these three different data sets, we simultaneously fit these, and then added the quarantine, our model predicted quarantine contacts with the parameters to fit also the quarantine contacts, and these are the fit we get. So the actual cumulative here is just a sum. We didn't fit the cumulative cases, we fit the daily cases, and I'm just summing up the daily cases. But we threw out 3 points in Hubei, which the data was there was problems with the data there. So this is why we just include a huge jump here, because there was some change in reporting characteristics. But this is our fit, and we can calculate then the effective reproduction number. The effective reproduction number without contact tracing is going to be the dashed line, and the blue is the actual. And so we see the impact of contact tracing, and also, again, the impact of the self-quarantine as the reproduction number sort of exponentially declines. And there's much more impact of the self-quarantine or lockdowns than there is the contact tracing, as again, we observe. And we can sort of observe this province by province. Okay, we fit here each individual province outbreak, and we see that there's this, we get this inverse, we recapitulate the inverse proportionality predicted from our final size formula. And when we color code it by the proportion of contact tracing, that really doesn't do any prediction for the final outbreak size. Again, these are our estimated parameters, but it shows this sort of the data, or our estimated sort of parameters here coinciding with our theoretical predictions. And this is I'm doing log scale on the y-axis, not log scale on the x-axis. Here's log scale, just to try to show when we look at the peak sizes in the outbreak, we do get more sensitivity for the large contact tracing provinces, the ones that had more contact tracing. So again, this bigger impact of contact tracing for peak size. So, we're aware that this was a very simple representation of the self-quarantine or lockdown. So, one criticism might be that we have a proportional to force of infection, which is like new incidents, but people don't, you don't see the actual cases until they're reported. So we could introduce a delay in our model through this reported, here's the reported cases, RFT minus so this is the daily reported cases. So if we have that, the self-quarantine is proportional to that instead of the force of infection, we can simulate the model. And what we see is that actually, in terms of our new proportionality constant, it's just going to, in order to get our original fit, we need to have a larger proportionality constant. And it can be theoretically shown that it's basically given by the baseline reproduction number beta times t times the original proportionality constant. So basically, if you account for this delay, you have to be even faster. And how much faster? Multiplied by the baseline reproduction number. Then we can try to also use, there was a lot of data from cell phone data, or mobility data from so the Baidu company, so we also fit with that, and it was in line with everything. So I'm going to skip that, but because I want to get to the next topic. Okay, so all in all, for China, we were able to look at the relative efficacy of contact tracing and lockdown. Now, for the U.S., we sort of had a different story because there's no contact tracing, like I said, or limited, and the reported cases were all over the place for each state. There was a lot of undetected cases. So we somewhat came up with a modified model here. So we still have, of course, this lockdown self-quarantine behavior that is proportional to the force of infection. Again, so we're going to use the psi parameter for that. We have some fatigue, which we call, or the lockdown exit rate alpha, we call it the fatigue, lockdown fatigue. And now for the US, again, there's a lot of undetected cases, unreported cases, and then we have the reported cases R, and then we're going to use mortality data also. So and for that, we use a gamma distributed delay from the time that they're infected to the time that they die, and that there's some data on that. Because it's decoupled, it's still an ordinary differential equation, but when we're fitting to data, we use a gamma distribution for that. And the hard part, though, is how do we get the proportion of cases that are reported? So this is the detection rate, or it's called ascertainment rate. So again, this, of course, Quentin, was dealing with this in his first part. So here for the US, it certainly was time dependent. And why time dependent? Okay, so I didn't really graph here the actual daily test as a function of time, but the daily test for each state, we're steadily increasing, but also very noisy. So the ascertainment rate is somehow a function of the testing availability. Okay, and so we assume that it's basically this saturating function, which seemed to fit well. And there's some mechanistic reasons for this. So basically, as the testing becomes more available, then the ascertainment rate goes up because more people, can take tests, and there's less delays. But it only does up to a point. So it saturates as, again, there's a limit on how fast the test can be done. So there's sort of this, if you think ecologically, there's this this capture rate factor, that this delay. And then also as the number of cases go, as the number of tests get to a certain point, once they become available, you start to see more of the increase in tests take, just because there's more infected individuals. So it's affected by both the availability of the tests and the demand from infected, or the actual number of infected people. It's sort of a combination of this, and I think, so we fit it individually to each state, and this seemed to be a good functional form. I should mention that we also did try to sort of make this a function of the reported cases, and so this, I can talk with Quentin about this. It kind of leads to this interesting functional form, but it didn't seem to work out with the data too much. So okay, we fit then each state using then the reported case data. So I'm showing cumulative reported cases on the left. The testing data is in the middle here. So the actual testing data is the red dots. We smoothed it a little bit and interpolated it to use in our functional form of rho of t here. And this allowed us to also, calculate the percent positive test, and we were able to sort of fit that pretty well. And then, of course, this is all hinges on mortality, right? You need to fit also mortality, because you can't get an accurate estimate of the unreported cases without the mortality, okay? Because mortality sort of is blind to unreported cases. It's sort of the thing that validates, validates everything. So we fit all of these three data sets, or use all three of these data sets, and these are some example fits that we got. And the goal was to characterize the lockdown, or one of the goals was to, and so we sort of had these broad characterizations based on our estimated parameters. So here's three states that are close to each other with different reactions. So we say intermediate lockdown speed, intermediate fatigue of the lockdown Nevada. So these are the characteristics. But California had a rapid lockdown, but high fatigue. And so we see the cases just don't really flatten after a while. And then Idaho here did have a little bit better response, rapid lockdown and intermediate fatigue. We can do for more states. Some states with high fatigue, like Utah, cases are almost increasing after a while. So the lockdown people just seem to be exiting the lockdown. And of course, New York was a notable example for a huge outbreak. And so they had a very slow lockdown. And so when we say slow, we talk about the lockdown, remember, it's proportional to force of infection. So it's not the time, the days since the first reported case that are necessarily important. It's the in terms of it's the proportionality with respect to the incoming cases. And so New York was very slow in terms of how many cases they were having in implementing their lockdown. So yeah, this is and again, on the left here, we have the reported cases and then our estimate for the true actual cases based on using our testing and mortality and reported case data. And we can do this aggregate for the US, of course, just in everything up. And we did that with the US. We didn't want to do it with every state because it's just so much data, but we did get some confidence intervals and everything for the parameters, which we obtained using this sort of bootstrapping Monte Carlo simulations. And we can also get the practical identifiability of each of the parameters that we fit. So R0 is one of the parameters. Psi is this lockdown factor. Alpha is the lockdown fatigue or exit rate. So these are the three control parameters. IFR is not a parameter, it's just the infected fatality ratio. We just estimated that. I0, of course, is the initial condition. And then again, these are not parameters, the true cumulative cases that we estimate, and then the true over the reported. So this is kind of like the amplification of the true over reported cases. I should mention, you know, I would think of this more when I say true cases as not including asymptomatic cases. So this is like only talking about symptomatic cases, because when you think we're using testing data, and so the people that get tested are more likely to be people that have some symptoms. So this is probably more of an estimate for the detection of symptomatic cases. But we could add asymptomatic into our, but then that's independent of the testing data, really. And we get, you know, for each state, we can get this. So, you know, in the last amount of time here, I just want to, so with this, we can get some correlation analysis. So with different quantities, you know, we find some significant correlations. One difference I just want to, you know, just in terms of, there was a study in France looking at the ascertainment, and it showed, so here we of course estimate the ascertainment or detection rate. So here we get that it's positively correlated significantly with the true cumulative case estimate, okay, for the states. This was actually opposite what was observed in France. And so the reason in France was because they had a good contact tracing effort. And so when the cases, the actual cases in the French provinces went up, the detection rate went down because it hampered the contact tracing efforts. But in the US, there wasn't really any contact tracing. So we get sort of an opposite, maybe because the more cases, the more people are sort of attuned to get tested, maybe. Again, we observe this inverse proportionality with the peak and final size cases. But in the US, we get straying, many of the states straying from the inverse proportionality relationship because of this lockdown fatigue. So states with high lockdown fatigue have higher cases than what they would have if they just stuck to their sort of lockdown, their lockdowns. Again, we can do sensitivity analysis. So possibly one thing, you know, one sort of policy implication when we were doing the sensitivity analysis in terms of the lockdown rate, or sorry, the lockdown, the time of the lockdown duration or fatigue. Okay, we noticed that basically there's this like critical length, we call it a population quarantine half-life, okay, so natural log of 2 divided by alpha of 30 days seem to work for bringing the cases down. So that's the time for 50% of the self-quarantined. So the amount of self-quarantined individuals depends on the R0 and, you know, the psi necessary for, you know, for that R0, but the length for 50% of the quarantine individuals to remain there, it seems 30 days independent of other model parameters. So because we can't have indefinite lockdowns, we know that. So just in future outbreaks, maybe, you know, this kind of knowledge of the 30-day sort of thing, and then sort of a you know, along with the gradual sort of letting out of the lockdown can be useful. And so in conclusion, you know, this is, you know, sort of some of our findings. I think I went through it pretty much. So, and I'm running out of time, so I'm just going to end here and with the references and acknowledgements. And if there's time for any questions, we can go ahead.
Thank you, Cameron. It's Jianhong Wu from York. Thank you for a very nice talk. I have a question about the quarantine, the fatigue rate.
Sure.
Is that fatigue rate is a constant? Is more populational characteristic, or is changing according to the trend of the epidemic, the epidemics?
That's a good question, Jiahong, and thanks for listening and asking. You know, we thought about different ways of doing that, but we went with just a constant. So it's just a constant exit rate. So I think, you know, I did see some people did try to make it a function of the, you know, amount of infected, because if there's low infected, people might just exit the lockdown. You know, this, of course, it can be like that, but it's maybe useful to have it just be a constant, because of course it's more simple and you can sort of think of it then as a, you know, as a fixed duration for the lockdown, or I mean, I like to think of it as, you know, in terms of this half-life, because things are not, you know, no matter what, you know, people don't exit lockdown in, you know, in one day, even if the government mandates it like that. There was numerous studies that, you know, people's actual behavior, it's a continuous, it's on a continuous spectrum. And so when you have a constant alpha, you can interpret it maybe like as a, you know, an exponential decay rate, you know, from the lockdown state, you know, and then you have sort of this interpretation of the half-life of the lockdown, you know. So I think it lends to better interpretation to have it constant.
Thank you.
So I think we both have a question, some question with Quentin. So Quentin, would you like to ask your question?
Okay, thanks. Yeah, I saw you mentioned the ascertainment rate. Of course, I'm very interested in about this this parameter. So I think you used this predetermined formula for this rate. So do you actually estimate something, or is it completely I mean
certainly
assumed?
We estimate the parameters K, A, N is just the population size.
Yeah, okay.
But K and A, and K0, we do this for each state, because each state was unique, and it's hard to sort of we weren't able to really confidently fix it for all state, you know, when we were trying to fit each state. Just so we did fit K and A for each state. And so A is like this is what you would call the half saturation constant. K is the maximum, sort of the maximum ascertainment rate. But the tau of T, of course, is data, right? This is the daily test, the smooth daily test. So just, you know, how ascertainment depends on the test. Yes, we fit that. Yeah.
Okay. So what you use is basically testing data.
Mm-hmm.
But and do you input mortality
too.
Also?
Yeah, and okay. Reported case data. So all, you know, basically, because the mortality data really is key, right? Because, you know, it kind of it doesn't depend on the ascertainment so much. It gives you more of an idea of the true cumulative cases, as opposed to the reported cases, which, you know, depends on the detection rate, of course. So
yeah, of course, it remains a kind of a difficult problem, because if you take into account mortality, then you have to estimate somehow the fatality ratio or something like that.
Yeah.
Not knowing the whole quantity of asymptomatic infections. So it's a very difficult problem.
I know. Yeah, sorry.
Okay, so just what kind of method do you use to estimate these parameters?
Well, yeah. So that again, that's why we used all three of those data sets, because the testing data, then can sort of be an extra sort of layer of data, which can for the mortality, because the mortality, yes, we can't confidently just use the mortality. It needs to be the combination of it, of all three for the U.S. at least. And we used just simultaneously fitting a weighted least square algorithm. And for the mortality, we tried to fix the case fatality ratio, but we got some nonsense fits. So we did also have to fit our case fatality ratio for each state, and it's but we bounded it pretty tightly to make sure that it was within the biologically reasonable for COVID.
Okay, thanks.
I have a very brief question about contact tracing here. What kind of model are you using? I didn't get it precisely to describe the contact tracing. You just did you mention that?
Well, so we didn't do it for the U.S. because we didn't have contact tracing in the U.S. because there.
Oh, okay. So in this part there was no contact tracing in this.
In the China there was, though, in China.
Yeah, sure. And so can you show us again the Chinese case? So you get the data for China, right? For the contact tracing in China.
Yeah. So we used, this is the total quarantine contacts. It's in the blue. So this is. Yeah.
Right. But what is your model again about that? What is the idea of the model again? So you amplify the. Can you show that? Because I don't see the.
Oh yeah. So this is the perfect case. So the psi here. Okay. So we assume that contact tracing. Okay. Again, mechanistically, it's proportional to the force of infection.
Right.
But so the actual contact tracing part for the susceptible, so the number of susceptible individuals, the rate at which they're contact traced is this 1 minus P over P times P. So Phi is the proportion per reported case that's traced. P is the probability of transmission upon contact. And so from this, we can get the susceptible contact traced. I see. But we also have the infected contact traced, and that's Phi times beta SI. And so we also add that. So it's basically a combination of the susceptible contact traced, or the ones that are not infected, were never infected, and the ones that were infected, because the data from China doesn't differentiate between the two. It only gives the total quarantine contacts, like both non-infected and infected. So we have to add them together, but we can still do that as a combination in our model.
No, that's interesting. And it seems to work perfectly fine with. Accordingly, you can fit the data very well with this this model, right?
Yes. So
it's perfectly it's consistent, at least, with the data. Oh, okay. Sounds very interesting. Is this part published already, or?
There's been a lot of delay in it. We submitted it first in June to some journal that strung us along, and then we had to resubmit somewhere in December. And so it's actually, a decision is in process right now, but it's on medRxiv. It's on.
All right.
Both works are on medRxiv.
Okay, that's very nice. Okay. Jean-Wong, do you have one more question?
Another question is about the final size. It's a index really tell you the outcome by the end. So this is more important than anything else. But the problem with this assumed the beta is constant, and in a pandemic, in COVID case, Its beta changes constantly from one count. So I'm wondering, I've been bothered a lot by this, is if beta is piecewise constant, is any way to really have an analytic closed form of the final size?
Well, again, I would argue, so we took it like, we sort of took another perspective on it. We tried to, okay, we think of beta as a fundamental parameter. It doesn't change. It doesn't, what changes is the population behavior. And so what changes is susceptible individuals going into the quarantine state. And that's determined by psi. That's the proportionality factor.
Beta will change for sure.
No, but it just depends on
beta contains the transmission, the probability to transmit the infection when you wear a mask or when you don't wear a mask, for example, it changes things. Well, and the contact contains also the number of contact per unit of time, and it changes also, of course, because people are more careful after some time.
Depends how you. I mean, it depends how you model it. I mean, so what we did was we said that this is what S represents, S of T, it's the people that are acting normally. What the people that change their behavior, they go to the SQ state. They're in the self-quarantine state. So we have a parameter for transitioning from the S state to the SQ state.
You get a lockdown after some time in China.
Yeah. Exactly.
So are you putting everybody in the anyway?
Yeah, of course. I mean, no, yeah, certainly. Like 95% of the population does go into that state.
I mean, there are some changes for sure. You cannot avoid that, right?
So Cameron's idea is really you transfer all this time variation to really to the time variation of the interventions, basically. Beta is the transmissibility kind of unit, fixed, is a biological characteristic. The contact is changing.
Yeah, so effectively, yes, effectively our transmission rate is changing, right? Because, if you think about it, where, the beta, the actual transmission rate is the beta SI over N, and so you're losing susceptibles. And so that's reducing when we look at, we can calculate, so just with that calculation, if we look at the effective reproduction number, it's exponentially decaying. And that's accounting for the fact that, in our model, we're saying, okay, we're keeping beta constant, but beta times S of T, of course, it's changing much more rapidly than a traditional SIR model, because we have this all these susceptibles go into the quarantine state. So it's just it's just a matter of a way to model it.
So for you, quarantine is accounting for the people who are more careful, basically, and
yes,
and were keeping their distance with other people. Okay, I see.
So as a peer, probably this Cameron's model will explain what I was trying to ask you, because beta is a true biological transmission rate, but beta multiplied by the 1 plus the psi is I call it practical transmission rate. Is Cameron also called it practical transmission rate?
Yeah, but this if you think about it, it's a good idea to use 2 compartments, but just another way. Probably it's also time dependent, because things are changing anyway. The rate at which you will move from one compartment to the next one will change also.
Yeah.
You get more people at the beginning. Nobody will go for quarantine, right? But
exactly.
So this is the same problem. So you transfer the piecewise constant from beta to psi and phi, or null.
Right.
So the fundamental issue is still if the parameter is piecewise constant, and would you still be able to do the final size calculation?
No, I mean, I don't think so.
I don't think so, Jean-Wang.
I know that because in the transition time, you don't know the transition time, and you don't have close form of the I in the transition time. And in the final size, I goes to 0 when t goes to infinity. That's
yeah, we're making a simple assumption on that form. Essentially, we're assuming that the psi is proportional to the force of infection. So, instead of saying piecewise constant, we're saying that, okay, the reduction in the effective transmission rate is basically goes down as the, it increases as in cases increase indirectly in proportion to that, and we it's almost like this is our phenomenological law. We kind of got the idea from reading, I think, like The Economist magazine, they call it exponential whiplash. Each country experience, when there's no cases, there's no motivation to go to lockdown, but as the cases rise, you get sort of this increasing, pressure to go to lockdown. So it's a phenomenological term.
Right. Okay. So I think we can, if there is no more question, I think we can stop here. And thank you very much, Cameron, for this very nice talk. And we will meet within 2 weeks with some new nice talk. And so have a nice day in North America. Okay.
Nice talk, Quentin and Cameron. Thank you for the very wonderful talks.
Thank you. Thank you for listening in.
Yeah, thank you all for listening.
Okay, so bye-bye, bye-bye, bye-bye.
Bye now.
Bye-bye.
Bye-bye.